Hello everyone, this is Chen from University of Arizona. I'm a PhD student at Department of Biostatistics. Today I'm going to present my recent work, Rapid Estimation of Node Significance in Weighted Bipartite Networks. In recent years, network-based approaches become very popular for understanding biological mechanisms. And the idea behind this kind of network analysis is that human disease is caused by perturbations within a small neighborhood of cellular network. So one main purpose of biological network analysis is to identify a perturbed neighborhood. In network science framework, this neighborhood is referred as communities or modules. In other words, this could be treated as a community detection problem. And there are a lot of well-developed community detection algorithms, but it is still a challenge to evaluate the significance of the detected modules. So here I want to present our recent work on how to fast evaluate node significance in disease modules. Talking about statistical significance, first thing we need is some non-model or non-distributions. So here I want to introduce a very flexible and a very popular generative model in network science called configuration model. Because it is not very clear what a configuration model looks like for weighted bipartite network, so we firstly derived its probability distribution. Here we start with a bipartite gene regulatory network with MTFs and N-target genes. And based on the soft constraint maximum entropy assumption, we want to maximize the entropy constrained with the degree distribution by introducing Lagrange multipliers to solve this optimization problem and with Boltzmann distribution. HW is the so-called Hamiltonian of the graph. Z is the partition function, which could be integrated out. Thus, we get an exponentially distributed graph. The maximum likely estimation for this distribution is to solve a system of equations like this. Many methods for solving this maximum likelihood are based on MCMC sampler, but here we choose to solve this system directly using spectral residual methods. This method is heuristic, so if we can provide a great initializer, this could be much more efficient than CMC. On my local machine, to solve a system with hundreds of TFs and thousands of genes only takes a few minutes. So here is a figure showing the strength distribution of the solved configuration model compared with subsampling network. So subsampling means we subsample a very large data set and constructed a lot of subsampled networks and plot its sampling distribution. It's obvious that subsampling is the most accurate norm distribution one can get when they have a very large data set. But most of the time, especially in biological experiments, sample size is very small. But we can see that configuration model is centered correctly, although overestimates the variance. This is because in configuration model, we only constrained on the mean of strengths but didn't consider the variance. This is a limitation of typical configuration model. It's worth developing a new model that can take both mean and variance into account. Next, I want to talk about how to evaluate node significance using configuration model. Let's consider modularity maximization-based community detection. The contribution of each node to the module it belongs to could be written as a formula here. So W is the disease network and N is the modularity expected network. If we want to evaluate the significance of some node I, we notice the discovered module structure and the expected age weight should be fixed. Actually, the only randomness comes from summation of Wij in the given module. Because Wij follows exponential distribution, so the summation follows hypoexponential distribution. And when the module is very large, we can use normal distribution to approximate hypoexponential, given the fact that the rate parameter theta I plus theta j is not very different for the same node I. In this way, it's not difficult to construct a statistical test based on the summation of Wij. Now we test our model on two bioportal gene expression datasets. One is ovarian cancer, the other is breast cancer. They are both large datasets, so we can use subsampling to construct some golden standard results. So figure A is based on the comparison between angiogenic ovarian cancer versus non-angiogenic ovarian cancer. We first construct gene regulatory networks for angiogenic and non-angiogenic separately. We use the state-of-the-art method PANDA algorithm to do this. Then we apply Opaca method to compare these two PANDA networks. Opaca is disease module detection algorithm based on modularity maximization. Then we apply our configuration model on the Opaca detected module to see if it improves the results. The bar plot represents the significance of gene ontology analysis. The higher the bar means the more significance. We can see from the plot the green bar Opaca overestimates the non-relevant goal terms. But configuration model and subsampling will reduce this noise. For cancer-relevant goal terms, in other words, angiogenic-related goal terms, configuration model and subsampling will do much better than Opaca. Figure B, we applied the same pipeline on breast cancer data, comparing estrogen positive versus estrogen negative. In this case, configuration model is not always working well. It overestimates the significance of housekeeping goal terms. We feel like this could be related to the problem of variance overestimation of configuration model. We will leave this problem for the future work. So some quick summary. Our method improved recovery of cancer-relevant goal terms in both ovarian and breast cancer. Here we use the PANDA network as an example, but technically our pipeline could be integrated with any weighted bipartite networks to evaluate node significance. We also noticed the problem of variance overestimation. This is unavoidable for large-scale gene regulatory networks, so it motivates us to develop a new configuration model to constrain both the mean and the variance in the future. That's all for my presentation. Thank you for your time, and let me know if you have any questions.